Okay, everyone, I'll go ahead and get started. My name is Greg Cooper. I'm happy to talk today as part of the lecture series in memory of Casey Brown, who was a great friend of mine. We were in grad school together and really wonderful person. And I'm happy to be able to do this and miss him terribly. But hopefully this lecture series is a little bit of reminds people of him and carries on in his legacy a little bit. So first, briefly, I'll give just a very quick introduction to myself. As I mentioned, Casey and I were grad students together. I did postdoc in Seattle and then ended up at a place called Hudson Alpha. And for those that aren't familiar, Hudson Alpha, which is pictured here on the right, this is an aerial view of our campus, actually a little bit out of date, but you can see what it is. We're a nonprofit genetics and genomics research center and education center located in Huntsville, Alabama. And you can see from what we like to claim is the world's largest model of a DNA double helix that runs through the heart of our campus here. Really, it's a little over half a mile long. We sort of wear our heart on our sleeve and what Hudson Alpha cares about. And here at Hudson Alpha, I run a genomics research lab, much like an academic department lab, where our focus is on approaches to discover genetic variants that causally contribute to human diseases. And to do that, we develop and apply a variety of genome scale technologies with a particular emphasis on understanding rare pediatric disease. So that's just a little bit about me and my background. So to get started, we're talking here broadly about human genetics with a particular emphasis on disease. And if you think about what human genetics really is at a basic level, it's the fundamental goal is to connect phenotypes, so things that we observe about human beings. And obviously disease is particularly important, but it could also be things like how tall you are, what your hair color is. And what we're trying to do is understand how genotype, so how genetic variation contributes to those phenotypes. And typically what this means at sort of a basic level is that you're looking for correlations, so non-random correlations between the presence or absence of a set of alleles. Our alleles are just different versions of chromosomes, of genes, of DNA sequences. And you're looking for that in different groups of people that either do or do not have a trait or have different versions of a trait, et cetera. So you're looking for correlations between alleles and traits or phenotypes. And ideally, of course, the correlations are important. That can be, you understand this allele A correlates to phenotype B, but we'd also like to understand the mechanism. So how does it work at a molecular level? What actually happens you have a change to a DNA, and then it leads to some molecular consequence, and then that molecular consequence changes cells or tissues or development or whatever the case may be, and that mechanistic process by which that change in DNA leads to a phenotype. That's obviously also of great value to learn, both for fundamental human knowledge, sort of basic research, and also for, as we'll talk about, a lot of clinical applications that emerge from that. So, and stepping back, so it's sort of looking at how we design human genetics experiments. And this is from a review that I had written along with Jason Dury many years ago, but it provides a big picture overview of what human genetics looks like. So again, typically at the top left here, we have people, this is blue and orange labeled people that maybe they have a disease or don't have a disease or whatever trait they have that's of interest. You perform some sort of strategy, and we'll talk about some of these in detail. So a genome-wide association study, or a GWAS, perform something called linkage, or do exome or genome sequencing. Again, we'll talk more about these. And essentially, these are strategies to discover alleles inside the people that you're looking at, and then to detect those correlations. So that in panel B here, you're seeing that these different loci, so these regions along the chromosomes in these individuals where you find some non-random correlation. So we're just labeling them A, B, C, and D, where you see this separation of alleles according to the phenotype that you care about. And then within each of those loci, in the bottom panel here, you might say that there's a number of different variants, so a number of different locations where you're seeing alleles that might be of interest, and they can have a wide range of functional categories. So we're showing different cartoons here. The panel A, for example, row A here has a traditional protein coding gene with a promoter, a 5' 3' UTR, coding exons, et cetera. And you can see there's a whole range of possible molecular events that variants can have. So we think about what the challenges are inside this big picture umbrella. The first is a statistical power issue. And this is really a major one that human geneticists have to fight with. And one way to phrase this, you might hear about something called multiple hypothesis testing. And to some degree, it is related to multiplicity, but as we'll talk a little more, it's really about thinking about prior probabilities. And I would encourage, there's a wonderful review from 2004, I believe, written in Genome Research by Manley et al. And it really talks about what the prior probability of something being true is the important thing. And in the scope of a whole genome, there are millions of variants. Typically, very few of those are expected to associate with a trait. Especially for rare disease, we'll talk about today. And so the prior probability that any variant at random is low, and that means you need a lot of data to prove that there was a real correlation there. And then there are haplotypic correlations. So if you're in common variant space, you might have heard of something called linkage disequilibrium, where alleles, and basically it all boils down to alleles tend to be inherited in segments where a large stretch of DNA comes as a unit. And so you have many alleles that give you the same degree of statistical correlation, even if only one of those alleles is actually functionally driving an effect on a phenotype. And then another challenge here at the bottom that you can see is molecular diversity. There's a whole variety of, as we talked about, promoters and UTRs and coding sequence and non-coding RNAs, et cetera. There's a lot of ways that an allele can alter molecular function, and in turn, a lot of ways for genetic variation to alter phenotype. And that can be a challenge because you don't know in principle. When you go to study a trait, you don't have any control or have any knowledge necessarily about what kind of function is relevant to that particular phenotype. So before we talk about how we discover variants that matter to traits, it's worth thinking about what are the types of correlations one might find. And the best way to think about this is to look at allele frequency. So this is how common an allele is. So if we have a variant that separates two chromosomes, you want to know, if I measure 100 random people or 1,000, how many of the two different alleles might you see? And phenotypic influence, which is to say that given that you have an allele that's associated with the disease, what effect size does it have? In other words, does it have a big effect where if you have the allele, then you have the trait, or is it small, or if you have the allele, then you have a very small risk of that trait? And so at the top right hand, so this, we all wish human genetics were like this, where you have variants that are very common in the population, meaning you don't need to see a lot of people. If you measure a small number of people, you'll see that allele multiple times, and that have a big effect. And so there are examples of this. So if you look at, for example, in pharmacogenomics, how people respond to certain drugs, like warfarin is a common blood thinning drug that there are common variation that have a very large impact on the right dose for that person. Other examples, which I believe we'll talk about in another lecture in this series, is looking at EQTLs, or expression quantitative trait loci, where an allele has a big impact on the expression level of a nearby gene. But for the most part, especially for disease, we tend to not see these, and the ones that exist have largely already been found. More commonly, if you move down this x-axis here towards you stay in the common alleles. So these are still highly frequent alleles, but the phenotypic influence drops. And this is what people call of common alleles small effects. And so many traits have this property where the trait has genetic influences, but any individual allele or variant has a very small effect. So things like height or cholesterol or diabetes, lots of traits are such that it's many small and many alleles of small effects that add up together to control whether you are to influence whether you have a phenotype or not. And you may have heard about things like something called polygenic risk scores, where people try to take advantage of the fact that while each allele by itself has a small correlation, but if you add up many alleles together, you can actually get a meaningful prediction about a trait. And of course, the other dimension, if you stay in this large effect on the right-hand side, move down the y-axis. So now we're moving from, we still have large effect alleles, but now we're moving to rarity. So these individual alleles are very rare in the general population. And this is where, and we'll spend most of the time talking today about, these are alleles that have a very large effect on a trait. So a Mendelian disease is a great example of this. So cystic fibrosis, for example, is caused by individual mutations. It's a recessive disease. So you have one mutation from mom, one from dad. And if you have those alleles, you have cystic fibrosis. And if you don't have those, you don't. So those are essentially almost deterministic in that sense of there's a strict correlation between the alleles and the phenotype. And there are, of course, other examples where it's not 100%. So there's something called low reduced penetrance. So mutations in BRCA genes, for example, that confer a very high risk of breast and ovarian cancer, but not necessarily 100%. But again, these are alleles that are very infrequent in the general population individually, But for the people that have them, it has a very, very large effect on their phenotype. So I talked about statistical power. So this matters on both axes here. So if you're moving down the x-axis, you lose power because the effect sizes are small. So if a given allele changes your risk of disease by 0.1%, well, then you're going to obviously have to measure it in lots and lots of people before you can make a confident assertion about that effect size. Just because if you observe 100 people and it's only a 0.1% effect, then you just don't see any difference. So you need thousands or tens of thousands of people to see that. And in a somewhat different way, but still the same effect of reduced power, is for alleles that are rare, you sequence 1,000, you identify 1,000 people in your study, and if only one of them has the allele that is impactful to disease, well, then you only see it one time in those 1,000 people. And in order to get it many, many times, which is, again, to get enough signal to overcome this signal to noise problem, you need to sequence or test lots and lots of people to see the effect. Even though the effect is large, because you have so few people that carry it, you have this, again, this fundamental issue of having enough power to find those correlations. And so now let's talk about what sequencing is. So as we'll talk about modern DNA sequencing, by modern, I mean by today's standards, we can very rapidly and efficiently identify most alleles, both common and rare, in any one person. In fact, the vast majority of alleles, we can pretty quickly identify those variants, those sites of variation, those alleles that they carry. And because of that, and because, again, human genetics kind of boils down to identifying these correlations between alleles and traits, they can be used in a lot of different study designs. So one is you can use sequencing in genome-wide association studies for common traits. You can do something which we'll talk more about called gene burden testing, where you're looking for individual genes that are enriched for rare alleles, each of which may be different from the other, but as a group, you study them. You can perform sequencing on small groups of individuals. So if you have a group of 5 or 10 people that you suspect have the same rare disease, you can just sequence them and try to find those correlations. And then, as we'll talk later too, about doing one-off experiments where you're looking at these so-called N-of-1 experiments, where you're looking at one patient or family that has a trait of interest and you want to find an allele that you think is contributing. So let's talk about the very basics here, and I'm going to gloss over a little bit, but it's worth understanding the basic mechanics of how we use sequencing to find variants. And it, of course, all starts with a person. And from that person, we collect genomic DNA. You know, a typical experiment here would be to get a blood sample and isolate DNA. You can get saliva samples, cheek samples, a range of different samples, but essentially you're trying to get genomic DNA from that person. And what you do then is you prepare a library from that. So this is a collection of fragments that you manipulate in such a way that they become sequenceable on whatever platform you're using. That library then, this consists of millions and billions of fragments of genomic DNA goes onto a sequencer. So this is a picture of a generic Illumina sequencer. Illumina has been a workhorse for a huge amount of human sequencing has occurred on Illumina machines of a whole different range of models and types. But basically, the machine then allows you to read out, for those millions and billions of fragments, to read out what their sequence content is. And here I'm showing you this is just 4 reads. And what we're saying here is, hypothetically, this person has an A allele and a G allele at a particular location. But of course, initially, that read that you have is just that one fragment. You have to place it in the context to understand where it belongs to and what that variant actually is. And the primary way we do that is through read alignment, which is to say that you take the sequence of those bases in each one of those reads and you compare them to, and very typically, and this is one of the most potent impacts of the Human Genome Project of the early 2000s, of the late 90s and early 2000s, when the human genome, quote unquote, was sequenced, was to generate this single common reference that allows us then to have a common frame of reference and a common way to look at reads from individual people. And when you do that, you get these alignments where you say, okay, now that read is not just floating off in space. I can actually give it a precise start and stop position inside the reference genome. And now I can register those 2 different alleles and say, okay, yes, I see that at this particular location, this person has an A and a G allele. So they're a heterozygote, they're diploid. And this allows you to do something called variant calling, which is to say that you go across all the reads that you've collected, you align them all, and you find all the positions, again, where it's a particular chromosome, particular position. You say, okay, in this site, this chromosome, this position has an A-G allele. And so we can say this person has an A allele and a G allele. And just to show an actual data slide here, there's something called an integrated genome viewer, where we're showing along the x-axis at the top here, this is just a short snippet of arbitrarily chromosome 4 here. And in gray are individual Illumina reads. And where gray is basically a site where that read matches the reference. And in fact, for any given person, the vast majority of positions are identical to the reference, so that's common. But you can see here in this middle column that's highlighted here, there are these Gs. So these are mismatches. These are sites where the reference is one base and the many reads include Gs, which are a mismatch to the reference. So you can see that this is a mother, father, child trio here, 3 different people. They're each heterozygous for the reference allele and a G allele at this site. And that's sort of what data, and I'm just not going to go into details here, but this is a lot of computation, obviously, that generates lots of information about individual variants. And what's important is they sort of all boil down to, as I've talked about, a chromosome, a position, a reference allele, an alt allele. And then there's a whole bunch of metrics about that relate to how the variant was called, why it was called, where it was called, and other information about it. And then genotypes for individuals. So you could put many samples. Obviously, you typically will study more than one person, where you have sample 1, in this case, their genotype is 0-1. So they're heterozygote for this variant. Sample 2 is 0-0, so they're homozygous. And there's obviously a lot of details I'm skipping over here, but safe to say this is the core mechanics. And when we talk about sequencing a person and finding variants or finding alleles, this process is what we're talking about. So now let's talk about how we actually use it. So I mentioned you can use sequencing as part of a genome-wide association study. And obviously it's very effective and actually increasingly now being used. Traditionally, however, sequencing is more expensive than arrays, which I believe you'll talk about that in a GWAS section. Arrays are a strategy where you just focus on a tiny subset, but a useful subset. So maybe a 500,000 or 1 million or 2 million locations of common allelic variation. And the advantage of arrays is that they're more cost effective than sequencing. And so to the extent that if you're considering doing a GWAS, to the extent that your sequencing is more expensive, then you might have to sacrifice sample size. So if your sequencing, let's just say, costs 5 times more than an array, if you have a budget to do 1,000 people on sequencing, that means you could do 5,000 on array. And you might decide that because there's this power issue where sample size is really important to overcome that noise, you might prefer the arrays for that because you can get much larger samples. And this is why, if you look historically, certainly the earliest GWASs were all arrays. And over the last 10 or 20 years, the biggest GWASs that have tens and hundreds of thousands of individuals are all powered by arrays because of the cost effectiveness. However, and what the rest of the day we're going to talk about is you're looking at rare mutations, rare alleles, this is where sequencing is necessary. Because the reason why arrays are cost effective is because they only target a tiny subset of all possible genomic positions. Now, again, that's a carefully selected subset that represents common allelic variation in human populations. But if you want to discover a rare variant, you need to do sequencing. And just the simple back of the envelope, there are 3 billion-ish positions in HD38. That's the common reference human genome. And each one of those, in theory, there are 3 billion possible point mutations, so that's single nucleotide variants. So if there's an A in a reference, then you can have a C mutation, a G mutation, or a T. So that's 9 billion possible alternative alleles. So this is where you can't really use an array to discover all of those, but sequencing can discover them. So now let's talk about sort of conceptually how that matters. So if you're thinking about a genome-wide association, and again, with this all in the context of finding these correlations between allelic presence or absence and a trait, and let's just do a simple cartoon of we have case alleles, we have cases here on the left and controls here on the right. And in this case, we found 8 blue alleles and 8 orange alleles in our GWAS versus controls where we have 4 blue and 12 orange. And then you might say, oh, look, I have this enrichment of the blue alleles in cases, and maybe that's an interesting correlation to me. And this is all done at a variant level because you're focused on common sites. This is 1 chromosome, 1 position, 1 pair of alleles. So this is the A is blue and G is orange, or vice versa. And so in this case, you might say the A allele at chromosome 1, position 1, is it heavily enriched in cases for this disease as opposed to the G allele. So when you GWAS, all your analyses are done at that individual variant level. On the other hand, when you're sequencing and when you're focused on burden or focused on rare alleles, you can't really do that because each site of variation is rarely present. So in this case, what you're doing is, and this is why it's called burden, is you aggregate a bunch of alleles at different sites of variation in some logical grouping. So a typical case would be to do this at the level of a gene. So if, for example, you were studying breast and ovarian cancer, and you were doing sequencing, you might say, give me all the individually rare alleles in BRCA1. This is a gene that's well known to associate with the risk of breast and ovarian cancer. And now each one of these blue alleles in cases is any rare variant in BRCA1. So each one of them might be different in terms of the genomic position, but as a group, their common feature is that they all affect the same gene. And this is the kind of signal you're looking at when you use sequencing to do these burden tests, is you're looking at the aggregate effect of many different variants, each of which the alleles are quite rare. So just to lay this out a little bit more, maybe a different way to view it is like this is a simple cartoon of your favorite gene somewhere in the genome assembly reference, where we have a promoter and a series of exons and introns. And you're going to find some cases, find some controls. You're going to do that sequencing that we talked about. And you're going to find mutations. And each one of these, so they're at different positions along this gene. You say in this case, we have 19 rare alleles and cases and 3 rare alleles controls. Again, each one is a different site or different variant. But as a group, you're seeing this aggregate signal. And then you do much like a GWAS, where you have to compute some measure of statistical significance for that association. You do the same thing for burden, only instead of looking at one allele, you're looking at one gene. So you get a p-value, which is to say a measure of how likely is that correlation to occur essentially at random, assuming there's no connection between the gene and the phenotype. And then you repeat that over all genes. So one typical strategy is look at all protein coding genes. And you can get, and so this is real data now. This is from a burden test that was done on a trait called ALS, or Lou Gehrig's disease. This is obvious. It's a phenotype that has a lot of interest for patients that are affected. And you can see here, this is what I'll probably talk about in a genome-wide association study, something called a QQ plot, which all of this is a measure of your p-values that are expected at random. So if you were to just randomly sample genes that have nothing to do with the trait versus what you actually observed in your actual cases and controls, you can see for the vast majority of genes here, the vast majority of p-values, the expected p-value for those genes matches what you'd expect. So the rank order is basically saying most genes don't have any signal for being associated. But as you increase significance here, and let's focus on this very top one here, SOD1, you see this p-value of 10 to the minus 8th-ish. And what that's saying is that that is unlikely to occur if you were testing random genes. So this suggests that SOD1 has a non-random enrichment for rare variants. And you can see here this first row here, where this is actual counts, and this is an illustration of what the kind of things burden will find, is in cases you're seeing 25 out of around 2,800 people, 25 out of 2,800 cases, about 0.9% in cases, versus controls used to have 5 and 6,400, so less than 0.1%. So this is an odds ratio of over 10. What that means is that there's a very large increase in risk if you have a mutation in SOD1. Again, there are many different mutations that contribute to this, but as a group, collectively, rare alleles in SOD1 are a risk for ALS. And in fact, this is one of the most commonly known genes. Of course, the flip side of this is ALS is a clearly complex trait driven by that can relate to many different genes. So even though if you have mutation in SOD1, you have a high risk, you can see the vast majority of cases don't have. In fact, a little less than 1% of people with ALS actually have a mutation in SOD1. So it's not anywhere near the only gene, but in fact, many genes can contribute to risk. And to think about that is, to push that forward is, what if you had a scenario where your burden actually was let's say in this case, we did this hypothetical study, we found 19 mutations in cases. And what if you had a study where you only had 19 cases total? That would mean that 100% of the people that you studied had a mutation in the same gene, as opposed to this ALS was more complex, where clearly it's many different genes. Just imagine a scenario where you're chosen your phenotype very, very carefully, and in fact, you enriched very strongly for one gene. And this is where this is a useful thing to think about, is so-called brute force resolution of Mendelian disease, which is to say that if you have a family or a series of families in which you suspect the same disease exists, and you believe it's Mendelian, and typically that means that it's rare. Mendelian diseases typically aren't common, and the few that are appreciable, so things like Down syndrome and cystic fibrosis and Huntington, have all been mapped. So typically you're talking about things that have very, very low prevalence in human populations. it typically must have some sort of very clear phenotype that is very unique. So if you take things like something like, I have a headache this morning, or my hair is brown, that's not likely to be Mendelian, just because that's not particularly distinctive. You're talking about very rare, unusual symptoms. Typically, we often think of Mendelian diseases, they are often, not always, but often very early onset, so in young children, et cetera. Then what you can do is just sequence. And this has become a very powerful mantra that we'll talk over and over again. You just sequence these folks and you look for variants that are grouped within the same genes and see if you can identify the source, the gene that relates to that phenotype. So one of the first demonstrations, to my knowledge, came from Jay Shenduri's lab, Sarah Ng et al. In 2009. And they studied a condition, in this case called Freeman-Sheldon syndrome, which is a joint and musculature problem. It is extremely rare. So they had 4 individuals, each from a different family. They also looked at 8 HapMap samples. In this case, HapMap samples aren't ideal controls because you don't know their phenotype, strictly speaking. However, it's a pretty good bet that they don't have this incredibly rare Freeman-Sheldon syndrome. And then what they did was they took these 4 cases and 8 controls effectively, and they looked for genes in which one or more of the affected individuals has a variant meeting one or more criteria, which we'll talk about. And then they compared that to controls. So here's the sort of top row of what they find, where we're moving here from heterogeneous to single gene. And by that, I mean, in this first column here, they're just saying in how many genes is there a non-synonymous splice site or coding indel. I'll just say that we'll talk more about something that looks like it changes the open reading frame, changes the protein sequence. And then they're saying, okay, this one individual has 4,500 genes, but if I intersect with this second individual, it's 3,200. And if I go to all 4 individuals, which is to say that if I assume all 4 individuals should have a mutation in the same gene, I have 2,500 candidates. So of course, 2,500 is not great, right? That's way too many candidates. You didn't really identify the gene. But you can do more than just look at non-synonymous. You can also apply filters to get rid of common variation. Again, we talked about, we're not doing a GWAS here. We're looking for the effects of rare alleles. So the first thing they did was to eliminate something called dbSNP, which dbSNP was, especially in the 90s and 2000s, was this universal generic resource where people that were doing variant discovery would deposit a dbSNP. So there are better tools now for looking at common variation. Nomad's a great example, but at the time, this was a good resource to get rid of variants that were common in human populations. And you can see right away a big difference. So in this monogenic category here, this 4 out of 4 column, we're already down to 50 genes. So we've gone from 2,500 down to 50. Then they said, okay, we have these 8 controls. Let's also filter out any variant that we see in the 8 controls. And now we're down to 20 genes. And if they combined the dbSNP and the 8 HapMap exons, they got down to 1 gene, which in this case, it was a positive control experiment. They knew the gene in this case. Nevertheless, it's kind of amazing that they were able to sequence just 4 cases and 8 controls and do some computation on the data, and bingo, you've identified the gene that's responsible for this Mendelian disease. It really is a remarkable demonstration. So now I want to take a brief side note and talk. There's a special category of rare mutations that are so-called de novo mutations. So de novo mutations are basically those that arise during sperm or egg formation, which is to say that neither mom nor dad have it, but their child does, because it's the mutation arose during the formation of sperm or egg, which led to that embryo. And the mutation, I'll use that word mutation, is not derogatory. It's in fact, every single one of us harbors lots of new mutations. It happens. It's a normal part of biology and genetics. And we typically have something like 70, 100-ish new mutations, every single one of us. It's just a natural byproduct of DNA replication. Most of those have little to no effect. Certainly mutations that are born that are inside a healthy baby, most of their mutations don't have an impact on them. But once in a while, a de novo mutation can alter a function and give rise to a phenotype. And this, in fact, is a key feature how you can have a scenario where you have a child that has a severe disease that neither mom nor dad has, and maybe there's no family history of. It can result from a de novo mutation, and that's the reason there's no family history, because that mutation just arose. And in fact, this gets back to the very roots of human genetics. So decades ago, when we couldn't sequence, when the first assays essentially of human DNA were something like this, it's called a karyotype, where you're just visibly looking at somebody's chromosomes under a microscope. And you can see here there's 23 pairs. where you have one set of chromosomes from your mom, one from your dad. And if you march through each of these pairs, if you look closely, you'll see here that this person has an extra copy of the 21st chromosome. This is called trisomy 21. This is what gives rise to Down syndrome. And this is not always, but often a de novo mutation, which is say mom and dad don't have Down syndrome. They are disomic for chromosome 21, but a mutation arose during sperm or egg that gave rise to that extra chromosome. Now that prevalence of trisomies is relatively rare. Point mutations occur at a much higher rate, as we mentioned, something like 70 to 100 per person, but there's still sufficiently few in any one person that becomes a very valuable candidate space to look at, especially when you have a severely affected child born to healthy parents. And so this becomes much easier to identify getting at this signal to noise issue. That's a small subset of things to look at that have a very larger prior probability for being causal. And ironically, in some sense, these things are also refractory to traditional approaches. So if you think about linkage, which you'll hear about in other lectures, family history studies and whatnot, don't benefit from that in this case because the mutation is de novo. It's not shared with any other family member. So a lot of traditional genetic tools don't work for traits caused by de novo mutations, but sequencing can. And so again, from Jay Shinduri's lab, Sarah Ng, in 2010, so about a year after the Freeman-Sheldon syndrome, they did a study of a phenotype that wasn't understood, something called Kabuki syndrome, and it's so named for this facial presentation. And what they found by looking at exomes was 7 of these probands had mutations in a gene called MLL2, and then they actually did follow-up sequencing on additional based on Sanger sequencing, so a more traditional, slower sequencing approach. They actually found 9 out of 10 of them had what looked like a rare bad mutation in MLL2, showing that this was likely the cause. And then they did a follow-up in a larger cohort, and what they found was about 2/3 of people diagnosed with Kabuki syndrome have a mutation in this gene. And nearly all of these were de novo, which is to say that the child had the mutation that neither mom nor dad did. So this, in fact, is a potent, de novo mutations are a very significant source of especially pediatric early onset conditions. So you combine these things together, if you look at this impact of sequencing, it really is tremendous on human disease discovery. So this is a review from Mike Bamshad in 2019, a little bit out of date, but the trend is really illustrated nicely here. So if you look at going back to the late 80s, early 90s with traditional approaches to identifying human disease genes, and then you look at it around exomes. So this is around 2010 is when exomes became technologically feasible. And you can see a very sharp rise in the number of Mendelian diseases for which we could identify a gene. And that was really driven by this ability to sequence handfuls of people with exomes, find genes in which they shared mutations, especially when you couple that to looking at de novo mutations, looking at comparing a child to his or her parents, it's really led to a massive increase, a really significant increase in our ability to solve Mendelian diseases. And at the other end of this is where I'm just looking at one person at a time. And so this becomes effectively a diagnostic tool. And if you think about rare diseases, Mendelian diseases, rare diseases individually are rare. So there's not a strict definition, but typically you might think of less than 1 in 1,000 or less than 1 in 10,000. But collectively, because there are so many different rare diseases, collectively they're quite frequent. So it's estimated something like 5 or 10% of people that have a rare disease. They all have different rare diseases, but as a group, they are common. Genetics are a big contributor to these. It's not necessarily all of them. There are viral infections. There are other kinds of reasons to have rare disease, but genetics are a big player to this. And as a result, by sequencing these folks and finding a precise molecular cause, we can give them a genetic diagnosis, a genetic explanation for the symptoms. And this can have a range of benefits, including one is recurrence risk. So the recurrence risk for a child with a de novo mutation is very different than, say, a recessive or dominant disease. It's much more accurate. It can at least, but be much more accurate prognosis in terms of, so if you're diagnosed with something like an epilepsy, but you don't know the cause of it, you don't necessarily know what to expect in the future, whereas a genetic diagnosis can help with that. You can do, again, with seizures as an example, you can do achieve better medical management. So different seizures have different mechanistic origins, and one medicine that works for one type of seizure won't work for another. And so you can make a better decision in some cases about drug treatments. And then support group and community building is really of tremendous value. Being able to connect for one family to connect to another family facing the exact same thing can be really useful to learn from one another and share experiences. And then, of course, thinking in the long term about future therapeutic research, future efforts to, whether it's drug therapies, educational interventions, et cetera, being able to identify these root genetic factors is really helpful to guide that future research. And in fact, in around the 2014, 2015 period, there was an explosion in sequencing as a diagnostic tool. And this is, again, a little bit out of date, but shows the trend nicely from 2019. And the key thing here is just looking at, these are all individual studies where they applied sequencing to diagnose a rare disease. And you can see here this number of positives, so the number of cases in which they found a variant that they think was related to the phenotype out of the total. And the bottom line is around 30 to 40% of individual patients can be given a medically relevant genetic finding. This is for neurodevelopmental disorders, which are a common category of rare disease. And so this has really become a very powerful diagnostic tool, this sequencing of individual patients, finding mutations that these rare alleles that are believed to be highly penetrant, and using that information in a clinically useful way. However, the flip side of that, and this is what I'll spend the next portion talking about, is of course that means if you're diagnosing 30 or 40% of rare disease cases, that means you're failing 60 to 70% time. Not failing in a derogatory, but just means that it doesn't work 100% time, and we'd like to understand why. And this has been true for other kinds. If you look at Centers for Mendelian Genomics, there was a large series of grants that really attacked Mendelian disease aggressively. And they found similar, where you see lots and lots of families in which you believe Mendelian disease exists, but can't find the gene. And what that means is that there's a lot of variants that probably aren't yet visible to us. And if you're, especially if you're thinking about doing research or a career in the future about doing genetic research, these are areas where there's a lot of work to be done. And there's a variety of strategies that we'll talk about, including annotation, data sharing, and different sequencing technology. So first, let's talk a few minutes about better variant annotation. And so I've talked about this a lot, that sequencing can reveal a bunch of variation, but there's this statistical power issue of, especially for a Mendelian disease, where you think it's really one allele that's driving it, one or two alleles that's driving it, and there are millions of alleles present in that person. There's very minimal prior probability if you pick a variant at random that it's going to be relevant. And so you have to come up with ways to sift through the information, to prioritize the things that you think are likely to be causally related to a trait. And the two big sources of information that one can use are, we have some knowledge, at least, about what the molecular function of different variants are. And we also know, in some cases, about their evolutionary history. And the first thing we'll talk about is non-synonymous variants. If we go back to our first Ng et al., the Freeman-Sheldon case, if you notice, the first thing they did is to focus on non-synonymous. So this is already, you're not looking at all alleles randomly, you're saying only those alleles that are non-synonymous, which is to say that they alter the protein reading frame. That's already applying a functional filter. And they also add another row, which I'm showing you here now, newly, is and predicted to be damaging. So this is taking not just all non-synonymous variants, but then doing extra computational work to say the subset of non-synonymous alleles that look like they probably are disruptive to the protein. And although they didn't need it in this case to solve the disease, where it might have helped, as you can see in this example where they said any 3 out of 4. So let's say that your phenotype wasn't 100% related to the same gene. It was only most people, like Kabuki case is about 2/3 are MLL2. So if that had been true, where it was only 3 out of 4, then they would have gone from 22 genes down to 3 genes by having this extra filter that says, give me things that are damaging, look like they're computationally at least look bad for the protein. And so this, generally speaking, why do we focus on non-synonymous variants? Well, one thing, we know that they have a functional effect. So you can say that at the very least, there's this immediate molecular consequence of changing the protein frame. And there's lots of data now, overwhelmingly so, that they are heavily enriched compared to randomly sampled alleles for disease, especially for severe Mendelian traits. Stop codons and splice height disruptions, so things that lead to truncation and elimination of a protein, are in a special category, because unlike missense alleles, where it's possible for a missense allele to have a range of consequences to protein, including no consequence at all, with stop codons, especially early on in a gene, you can say, yes, that no protein will be produced from that particular copy. So that's in a special interpretive category. And the bottom line is that this prior probability is much higher for non-synonymous variants than for randomly sampled alleles. And this is why exome sequencing, and exome sequencing was originally conceived because it was a way, especially when sequencing was more expensive, you couldn't afford to sequence all of the human genome, so all 3 billion bases. But the exome, which are all protein coding exons, are about 1.5 to 2% of all the genes. That was much more cost effective. And the reason it works is because of that enrichment that it provides for disease effects. However, you still have to keep in mind that most non-synonymous variants don't really have an effect on disease. And so, as we talked before about this damaging issue, about we want to find those non-synonymous alleles that are more likely than a random non-synonymous one to actually affect a protein and give rise to a disease. So there are a lot of ways that we use information about individual non-synonymous variants. So biochemistry, things about like the nature of the amino acid. We understand at some degree the nature of protein structure and folding, binding site, et cetera, and conservation. So this is a pattern that is very important to lots of looking at variants, looking at their evolutionary histories. Typically in vertebrates, with proteins, we're talking about vertebrates. So humans to other mammals, of course, but chicken and fish and other kinds of vertebrate animals. And there are two different ways to go about this. One is a model based, where you sort of set up a hypothetical definition of what a mutation that causes disease and doesn't cause disease look like. And another is where you train a model. So things like AI approaches, where you're saying, here's a set of variants that we know cause disease, and here's a set that we know don't cause disease, and learn what the properties are that separate them. So there have been many examples of this. This is one of the earlier ones, a program called SIFT. And this is based on just conservation. So at the top here, they're comparing a protein across many different species, and then they're measuring the conservation, where the height of these letters. So in this TBS here, these are positions in this protein where it's only TB and S that are observed. And so you see this high degree of conservation. And the flip side of that is that you predict that most mutations here are likely to be bad. Whereas these sites here, where you see over evolutionary time, you see many different amino acids. The flip side is that very few of them are likely to cause a phenotype. Very few of them are likely to be deleterious to the function of that protein. Another example, this is more of a trained model, is something called Polyphen, which is a popular prediction tool where they feed a bunch of known benign and known pathogenic mutations, and they learn. This is where you feed it many different annotations, and then you build a model that allows you to separate out pathogenic and non-pathogenic alleles. And just one example, this is a generic way of talking about this that I'll show a couple times here, is if you compare mutations in a random healthy trio, so this is just looking at a mom, dad, child, this is all the mutations that you see in those three people, versus all the mutations that are in ClinVar, which if you're not familiar, ClinVar is a federal database where lots of labs that study human disease deposit their variant. When they find a variant that they believe is causal for a disease, they will annotate it in ClinVar as being pathogenic. And if you compare, so this is looking at SIFT scores now, where low scores are sites where SIFT says this looks bad to a protein, and high scores are sites where it's likely to be tolerated. You can see that ClinVar mutations that are pathogenic, so things that are believed to cause disease, are heavily enriched compared to the vast majority of pathogenic mutations have this SIFT score of 0, as opposed to if you randomly sample variants from a trio, you can see most of them, in fact, are shifted towards the right hand or shifted towards higher scores. Now, again, there are plenty of randomly sampled mutations that have this low SIFT score, and there are also some disease-causing mutations that have this high SIFT score, but you can see this separation where you're getting information content, you're prioritizing more effectively than you would at random. So now let's take a step back and talk about so non-synonymous variants have tremendous value, and again, that's the very basic premise of exome sequencing. However, there is this other 99%. In fact, the vast majority, 99% of all variation, is outside of protein coding genes. And what about that? In fact, this is something that Casey and I spent many years together in grad school. This is a question that really drove and excited us. And just like for coding variants, non-coding variants benefit from a similar kind of annotation strategy. So one is comparative genomics, evolution. Unlike proteins, where you're looking at vertebrates, in non-coding space, we're typically looking only at mammals, or even narrower, because most non-coding material is not survived selection over the course of vertebrate or farther evolution. And we, much like non-synonymous, we can also talk about function. In this case, it's different kinds of function. It's not amino acid biochemistry or protein biochemistry. It's gene regulation. So these are sites, we're interested in sites that actually change how and when genes are expressed. And these could be things like transcription factor binding, chromatin modifications, et cetera. So this is just one example of, unlike the protein conservation, here we're looking at nucleotides, so non-coding conservation, where we're looking at human sequence compared to sequence from a bunch of other different mammalian species. And you can see here that the human sequence is identical, with a few exceptions, to that same stretch of DNA in all these other species, which suggests that these sites encode for important functions. And if they were to be mutated, they might be more likely to have, give rise to a phenotype. And this can be done at the regional level. So you can look at a window. So if you read the original mouse genome paper, which I would encourage you to do, it's a really wonderful paper. They did this 50 base window and you're looking for conservation across a window. But you can also look at individual sites. And in fact, the richer your sequence alignment gets, the better your ability to look at individual sites and say, this site has a given conservation score. It's higher than this one, is intermediate to this one, et cetera. And much like CIF scores, you see the same separation with just using nucleotide conservation. So here we're looking at ClinVar pathogenic versus randomly selected trio mutations. And again, in this case, higher scores mean higher conservation. So we would expect disease-causing mutations to be at conserved sites. And in fact, that's what we see is this large shift towards high conservation for pathogenic mutations. As I talked about, there's also a whole variety of nucleotide level functional features, again, mostly to do with how transcripts are regulated and processed. And there's a bunch, and because there's such a huge diversity of ways that can happen, there's a bunch of assays to do this. And I'm not going to talk about details. I encourage you to go to the ENCODE project. This is an older ENCODE project paper. There have been a series of papers since then, but the ENCODE was focused on finding functional elements outside of coding exons. And this is just one cartoon of showing data saying, oh, look at this genomic location. There's a bunch of different measure of functional activity here. And what ENCODE did was do huge numbers of experiments. So this is just a tiny sample of all the different cells and proteins and other things that they measured. And this gives rise to the next sort of challenge is how do you handle all this data? And in fact, so we know that evolution is useful. We know that function is useful, but how do you compare the two? What is the value of different functional bits? So a promoter versus an enhancer versus a splice site, et cetera. And then how do you handle combinations and just the sheer amount of data that goes in that's available to look at annotations of human genomes? And we don't have time to get into it, but there's a bunch of different, and this is a very active area for development. But over the past 10 years, we've seen a lot of work towards building these so-called integrative approaches where you try to combine many different annotations through a machine learning or other statistical framework, again, based upon some modeling or training about what you believe pathogenic and non-pathogenic mutations look like, and come up then with a single score that combines the input of many, many different features. And this is, again, an area of where there's been a lot of progress, but this is also an area where there's a lot of active work. So there's still, we could always get better at being able to predict which mutations are likely to give rise to a phenotype. And just as one other example, getting back to this trio versus ClinVar again, this is something called CAD scores, which is developed between my group and Jason Dury's group about 10 years ago. And again, this is one of these combined where it looks at any mutation anywhere across the genome, both coding and non-coding, and it comes up with a single score. And you can see here again, for ClinVar mutations, they tend to have CAD scores above the high teens and 20s. Whereas if you randomly sample alleles from a healthy trio, the vast majority are below 10 here. Again, they're not deterministic. So just because mutation has a high CAD score doesn't mean it's pathogenic. And just because it has a low score doesn't mean it's not pathogenic, but you're separated. You're more effectively prioritizing than at random. Okay, so now I'm going to have two very short stories about the other two approaches to do a better job at this. And the first one is data sharing. And it sounds kind of simple, but this is hugely important. And for rare disease especially, this comes down to the fact that many conditions are extremely rare. So imagine, for example, a prevalence of less than 1 in a million people that has a particular disease. Well, that if you sequence literally every person in the United States, you might only get a couple hundred that have that trait. And obviously, no individual labs are sequencing every human being in the United States or the world, et cetera. And so there has to be a way for labs that focus on rare disease genetics to share data, because you're typically only going to find, any one lab will find only one patient, only one allele in any given gene. And it turns out this data sharing tool, this sort of Craigslist model, there's a tool called GeneMatcher. There are several like this that actually communicate with one another, where this has really been very transformative for rare disease, because all it does is, instead of having to share somebody's whole genome, you just say, I'm a researcher, I found a variant in gene X. And then if anybody else anywhere else in the world gives their email address and says, hey, I also found a variant in that same gene, then the system says you two should talk and see if there's a match there and follow up. And this has turned out to be massively important for rare disease genetics. So this is around 2015. This is from a review from the developers of GeneMatcher. And you can see here, since 2015, since this matching system was developed, there's been a massive increase. So hundreds of genes have now been discovered that would not have been discovered by individual labs because they only saw one case. But with GeneMatcher, you go from one case to 5 to 10 and 20, et cetera, just by having this ability to share this data. And this is actually a credit in many ways to the sort of early founders, the early human genome days. A very important principle was openness and data, public data sharing. And that's carried forward and had a huge impact on rare disease and continues to have a big impact. Okay, so for just the last few minutes here, I want to talk about a technology story. And this is long-read genome sequencing. And in many ways, the arc of genomics is really a lot about technology driving so exomes, for example, were a major technological leap that unlocked lots of discovery. And more recently is the so-called long-read sequencing. And to place it in the context here, go back to our original cartoon of the basic workflow of how we discover variants, which is to make fragments, fragment libraries, put them on a machine, generate reads, align those reads, call variants. Well, read length matters, and it's because of this nature where you're chopping up a person's DNA into millions of fragments. And typically Illumina, which has been this workhorse for a long, long time, your typical experiment is 150 base pair reads from both ends of a 300 to 500 base pair fragment of DNA. And typically what you do then is, again, we have to align those to the reference genome. And this is a little like putting a puzzle together. And unfortunately, that means small pieces, so these 150 base pair chunks can often look very similar to one another. And to place this into context here, let's do a simple cartoon of, we have genomic DNA for a person, we make a library, and our challenge once we get those reads is to put them back along the genome where they belong. But now let's give a ever so slightly more realistic version of this, where now I'm putting these red bits in, and what these red bits are the same sequence repeated over and over again, which in fact is a very common feature of human DNA. And then when you make the library visualize it this way, you now have these 3 red reads. And for each one, you don't actually know where it comes from because they're all the same sequence. Essentially, that puzzle piece all is the exact same color, and you have no idea where to place it. In contrast, if you make the fragments longer, then you span those repeats. So now we have these 2 long fragments, and I can confidently put this left one goes to the left and this right one goes to the right. Because the read length is much longer than the repetitive sequence, I can place that repeat into context. And this is the real power of long-read sequencing. And in fact, it actually does a better job. So the one way to do this is do these benchmarks against, this is data from our clinical lab here. And they're just looking at, so this is one version of this called PacBio HiFi that is very effective. These HiFi generates reads that are 10 to 20 kilobases in length as opposed to 150 bases. And you can see by this measure here, this details aren't terribly important. This F1 score is just a measure of the balances, both specificity and sensitivity. You can see we do a much better job with long reads. So we're getting much higher accuracy for calling variants than even from a gold standard clinical pipeline based on Illumina data. But where long reads really shine is to pick up variants that you couldn't see at all before. And this is just one example that my group published some years ago, but there have been lots of other examples of this. And what we found here was about a 7 kilobase insertion. And what was unique about this insertion is that it had these duplicated sequences in it. And in particular, at the ends, it has something called a target site duplication of 119 bases. And why this matters is that if you look inside this 119 bases, it includes a coding exon of this gene called CDKL5, which is associated with seizures. And this resulted in frame shifting the transcript and leading to a loss of function. And in this case, the insertion was actually mediated by something called an L1 event. And L1s are mobile elements. They're like those red bits where there's many, many L1s spread across human genomes. So with short reads, you can't identify this insertion because the reads map to too many locations. But long reads are able to see it. And in fact, L1s, and there are lots of other elements. So you can see here, this is just annotation of repeats. So ALUs, for example, in fact, ALU sequences are about 300 base-ish long repeats that are present that are about 10% of human DNA. So it's, in fact, human DNA is incredibly repeat rich. And so this, in fact, has a huge impact on our ability to align reads. And so this is where this new sequencing technology is unlocking variants that we couldn't see before, and is really an exciting area where there's a lot of work being done for the future. So just to conclude, in general, large-scale sequencing has really transformed genetics, especially for rare disease. In many cases, you can rapidly go from a genotype to a phenotype, and that has a profound diagnostic benefit for individual rare disease patients. But there's a lot of work to be done. And again, this is where if you're thinking about, if you're interested in genetics, you're excited about these, there are lots of areas to do things better, obviously. Looking at how do we annotate, how do we interpret, how do we understand variation, especially non-coding. Data sharing is really critical. Again, this has been, and can be challenging. There are some ethical and legal issues in addition to the pragmatic issues of sharing genome data sets tend to be large, but tools for data sharing are really critical. And then obviously, again, if you look at the long-term arc of genomics, it's about sequencing and computational technologies that continue to improve and provide a bit better ability to see and understand genetic variation. So with that, I'll just put a picture here of Casey. This is him trying to kill me on the side of Mount Rainier. He was, I encourage you to look him up. Casey, we had a lot in common. A lot of the stuff that I've talked about today, we conceptually and spiritually had a lot in common. We spent a lot of nights in grad school talking about evolution and function and how do we understand genetic variation. And he was a wonderful scientist, but he was an even better person and a great friend. And so I miss him and hope this lecture series is a little bit of an homage to him. And with that, I'll say thank you all and conclude.